Hello friends, welcome to the another video from BioShots. In today's video, we'll talk about DNA fingerprinting principle. So first of all, what is DNA fingerprinting? DNA fingerprinting is a technique to determine the nucleotide sequence of some area of DNA which is unique for each individual. The technique was developed by a British geneticist Sir Alex Jeffrey in 1985, and in India the technique was introduced by Lalji Singh and Kashyap. Now the principle of the DNA fingerprinting, it is based on the fact that there are some 3 × 10 raised to power 6 base pair differences in between the genomes of any two organisms. So if we compare genome of any individuals, there will be at least 3 × 10 raised to power 6 base pair differences. Now some of these differences lie in the repetitive DNA. Now what is repetitive DNA? Now significant, we know that significant that portion of our genome comprises of this repetitive DNA. In repetitive DNA consists of the sequences that are repeated many times. So repetitive DNA comprises of sequences that are repeated many times. So this repetitive DNA is of many types depending upon the position of the repetitive DNA and the length of the basic unit. And then these repetitive DNAs are usually present on the non-coding region of the autosomes. And as far as their types are concerned, we know repetitive DNA, when the same repetitive sequence is present at the same site, then it will be called as the tandem repeat. For example, in this example above, you can see that same sequence is repeating at the same site. For example, so therefore it will be called as the tandem repeat. For example, you can see here that this sequence which I am highlighting, that is ATTGC, this is being repeated one after another at the same time. And this sequence ATTGC is called as the basic repeat because this is the sequence that is repeating, for example, in this case 3 times. In example second below, you can see that the same sequence is repeated, but the basic unit is interspersed by the non-repetitive sequences. So the portion highlighted in green is the non-repetitive sequence. So here the same sequence is interspersed by the non-repetitive sequences. Hence this type of repetitive DNA is called as interspersed repeat. So interspersed repeat is the one which is interspersed by the non-coding regions. Now next we will find out what is this basic repeat. Basic repeat, as I told you, it is the sequence that is repeating many times. So those basic repeats which are up to 60 base pairs long, those are called as the satellite DNA. Now satellite DNA has been named so because they appear as a separate band from the bulk DNA during what is called as the density gradient cesium chloride ultracentrifugation. So when we do centrifugation of the DNA, 2 bands will appear. One will be called as satellite DNA, that will represent this repetitive DNA, and other will form the bulk DNA. Now satellite DNAs are further classified into several categories depending upon number 1, the length of the sequence, the GC content of the satellite DNA, as well as the number of the basic repeats. For example, if the satellite DNA is between 11 to 64 base pairs long, then it will be called as the mini satellite. So any satellite DNA where the basic repeat is 11 to 60 or 64 base pairs long, then it will be mini satellite. And the satellite DNA, when basic repeat is less than 10 base pairs long, then that type of satellite DNA will be called as the microsatellites. They also differ with respect to the number of the basic repeats. Now, mini satellites and microsatellites, they are named otherwise as well. For example, microsatellite is often also called as simple tandem repeat, and the mini satellites, they are also called as VNTRs, where VNTR stands for variable number of tandem repeats. V for variable, N for number, T for tandem, and R for repeat, variable number of tandem repeats. Now let us talk more about these VNTRs. VNTR, their number varies from a person to person. For example, this number can range from 2 to 200. A person, some will have at a specific locus 2 VNTRs, 2 basic repeats at a VNTR loci, in others there will be anywhere between 2 to 200. So in human beings, there are some 60 loci in their genome where VNTRs are found. Those are called as the VNTR loci. Now if we compare the VNTRs of two organisms, so if we compare two VNTRs of two individuals at all the VNTR loci, 60 VNTR loci, what we will find is that not all the VNTRs will be same. Some will have, there will be definitely some differences with respect to the number of the basic repeats at a particular VNTR. So this represents that we are different with respect to the VNTRs. VNTRs are the flanked by the conserved sites called as the restriction sites. So all the VNTR loci, they will be flanked, at their ends will be restriction sites. So therefore, if the same restriction endonuclease is used to cut the DNA or the VNTRs of say two different individuals, it will result in production of the different sized fragments. So fragments of different sizes will be obtained depending upon the number of the basic units present at each VNTR loci, which can range from 2 to 200. So as these different sized fragments are produced, therefore different organisms will show what is called as the polymorphism. Now this type of polymorphism is called as restriction fragment length polymorphism, because the same restriction endonuclease will result in production of different sized fragments. This is the basis of the DNA fingerprints, that when same restriction endonuclease is used for the two different individuals, they will have different sized restriction fragments. Let us study this example. So here it is, suppose a VNTR locus, and in this VNTR locus is the tandem repeat. This tandem repeat is marked by this highlighter. This is the basic repeat, and here as you can see, the basic repeat is 11 base pairs long. It is the 11 base pairs long basic repeat, and it is being repeated in one individual by some 4 times. There are 4 copies of this basic repeat. Now another individual will have different number of this basic repeats at the same locus. So different individuals, they can have different number of basic repeats, and they can be anywhere between 2 to 200. So therefore, when the restriction endonuclease applied to them, they will